So this class is on drug metabolites, regulatory perspective and safety testing. This is part of the syllabus for modern bioanalytical technique, where we will be discussing what is drug metabolite profile and what is the existing regulations for this and how to comply with the safety testing aspect of it. So drug metabolism is about biotransformation of drugs. Biotransformation of drugs basically happens in two phases. One is phase 1 and phase 2. The objective is to clear the drug from the body either in forms of metabolites. So the metabolite which is formed because of the biotransformation of drugs, it may possess a similar or unique safety profile relative to the parent drug. That is, the safety profile it may be similar to that of the parent drug, or it may have different profile. Usually the phase 1 metabolite are more likely to be chemically reactive, and therefore they are basically more considered to be pharmacologically active. This is because this phase 1 metabolite involves specific chemical reactions like dealkylation, alkylation, oxidation, reductions. So it forms active metabolites as compared to the phase 2 metabolites. The phase 2 metabolites basically they render a compound more water-soluble because of the conjugation to polar moieties like sugar, glucuronides, etc. So the phase 2 metabolites are less likely to be pharmacologically active. Nonetheless, if the pharmacologically, these phase 2 conjugates are found to be active also, their safety also needs to be tested. The metabolites formed by biotransformation of drugs may have affinity for off targets. That means they may bind to those receptors which may produce undesirable effects or unintended effects. So that may lead to adverse effect or toxicity. That is why the metabolite once formed, their safety needs to be assessed. So their safety can be assessed by the use of toxicity studies. So by doing that, the safety will be evaluated, and that will be establishing the suitability for further clinical evaluation of the drug and the metabolite. So, however, demonstrating that a metabolite is inactive, suppose we demonstrate that the metabolite is inactive or it is not having any pharmacological effect, that does not mean it may not have any toxicity effect. That is why the metabolite safety should be demonstrated in terms of toxicity evaluation, and for that in general non-clinical toxicities are done first. So once the non-clinical toxicities are done, then these are extrapolated for in vivo human conditions. That means these are extrapolated to human conditions or human toxicity or toxicity in vivo. But that may not be always true, because the metabolites which are formed under non-clinical conditions may not proportionately form in human conditions or in vivo conditions. Basically, metabolite which is formed in humans may not be formed in animal test species. So that is a kind of qualitative disproportion between human and the animal metabolism. But this kind of qualitative disproportion is relatively rare. What is more common is quantitative disproportion. That means the metabolite that is formed in human is formed at a higher level compared to the animal test species. So this is a more common phenomenon. So this shows that the metabolite is qualitatively or quantitatively different as compared to the non-clinical species which was used for the analysis. So there is discrepancy between the level of metabolite formed in the non-clinical model and in vivo. That is why these categories of metabolites or this kind of metabolite are called as disproportionate drug metabolite. Identification of the disproportionate metabolite should be done as early as possible in the drug development process, because their identification in late stage of drug development will be adversely affecting the drug development process, and that will also add up to the failure. That is why the disproportionate metabolite should be identified at the early phases of drug development process, and if the disproportionate metabolite is present at more than 10% of total drug related exposure at steady state, then that is a concern. So, in that case, those disproportionate metabolite should be considered for safety assessment. So, this disproportionate metabolite will be evaluated for toxicity studies, and there are two approaches basically adopted for this toxicity evaluation. In one case, the disproportionate metabolite is identified and tested in an animal species routinely used in toxicity studies that forms the metabolite either equally or in higher amount as compared to human. So, identification of a model or animal species in which that metabolite is formed either in level which is equally or greater than human is critical in this approach. In another approach, the metabolite is synthesized and then directly tested in animals. So, this is preferred in conditions where the likely or the suitable animal species or animal model is not available for metabolite analysis. Then we can make synthesis of this metabolite and directly test that in animals. There are limitations in using synthesized metabolites for this toxicity evaluation, because when we are using synthesized metabolites for toxicity evaluation, you are directly administering the metabolite to the animal species, and while doing that you are it becomes different from the clinical situations, because the metabolite itself is will be undergoing metabolism, and that is why it may not reflect the actual clinical situations which you are expecting to get because of the administration of the drug. Besides, this may also give rise to new and different toxicities. Nevertheless, identifying and evaluating potential toxicity of metabolite is necessary for safety evaluation, which will be acting as a guiding force for further clinical evaluation. Now, coming to identification of metabolite. A metabolite profile should be identified at different stages of drug development process. Basically, it is preferred that the metabolite profile is known in the early stages of drug development. So, for that purpose, basically several in vitro methods are used, like use of liver microsomes, liver slices. Hepatocytes can also be used as a in vitro model for determining this metabolite profile. So, once finding this metabolite profile in vitro, they should be correlated to the in vivo metabolism. So, if there is any discrepancy, that should be noted. Besides, in vivo metabolism in human also should be done early, because this will be helping identification of disproportionate metabolites. Analysis of metabolites is key to the metabolite profile establishment. So, finding a valid analytical technique which will be establishing the metabolite both qualitatively and quantitatively is critical to the metabolite profile determination. So, the in vitro metabolite profile can be poor predictors of in vivo circulating major human metabolites. This is because sometimes the exact structure of major human metabolite cannot be clearly elucidated. Also, a standard metabolite cannot be easily synthesized for use and for evaluation. So, in those cases, non-traditional approaches needs to be adopted to determine this metabolite profile. Several approaches have been used, which are basically fit for the purpose methods, including this mixed matrix LC, MSMS, peak area comparison across species. Then there are NMR based techniques also that are used, and besides that, bioanalytical methods are used, which use basically radiocalibrants for this purpose. While designing a non-clinical study for this disproportionate metabolites, the following things needs to be taken care of. First of all, the similarity of the metabolite to the parent molecule should be checked. then the pharmacological or chemical class of the metabolite or the disproportionate metabolite should be established. Its solubility should be established, then its stability in stomach pH should be established, especially when or if this metabolite is supposed to be administered orally. Then phase 1 versus phase 2 metabolite pattern is also needs to be tested. Then what is the relative amount detected in human versus the amount detected in animal should be compared. The toxicity studies which are recommended for the disproportionate metabolite include the following. First of all, the general toxicity studies should be carried out as per the ICS guideline. The toxicity studies should be carried out at levels which are either equal to that of the human level or multiple of the human level of exposure. And while doing this kind of evaluation, it should be kept in mind that the route of administration should be the same as that of the drug itself. If the route happens to be oral, then it is important to remember that the stability of the metabolite in the GI tract needs to be established, because this will be affecting the bioavailability of the metabolite or the systemic exposure of the metabolite. So, in this case, if the metabolite is to be given orally, the toxicokinetic data of that needs to be established and related to the systemic exposure and the toxicities observed. Besides general toxicity studies, genotoxicity studies should be carried out through in vitro assays to detect any point mutation and any chromosomal aberration. Besides that, if the drugs are intended for use in people or the population that include women of childbearing potential, then embryo fetal development toxicity studies should be carried out. Besides that, other reproductive toxicity studies can also be carried out case by case basis depending upon the results of general toxicity studies and the results of embryo fetal development studies. For drugs which are intended for chronic use or recurrent use, if the carcinogenic potential of the metabolite cannot be adequately evaluated during the carcinogenic studies conducted with the parent drug, then the disproportionate metabolite should be separately studied for carcinogenic studies. Let us discuss the case study of this azilsartan. Azilsartan midazolam is the prodrug which is converted to azilsartan in vivo. Azilsartan itself is metabolized to M2 and M1. M2 is the major metabolite and M1 is the minor metabolite. The level of the systemic exposure of the major metabolite, that is M2, is 50% of that of the azilsartan, and the minor metabolite M1 level is less than 1% of that of the azilsartan. Both of these metabolites do not contribute to pharmacological effects of the drug. That means they do not bind to the same receptor as that of the azilsartan, but at the same time their affinity for other targets or the off targets was not available. That is why it is necessary to establish the safety and toxicity of these metabolites. Since AM2 is present in greater than 10% of that of the azilsartan, it was necessary to evaluate the safety and toxicity of the M2. So, M2 was studied in 13-week rat and dog repeat dose toxicity studies and in reproduction developmental studies and found to be safe. Although the M2 tested positive in the Chinese hamster lung cytogenetic assay, it was found to be negative in other genotoxicity studies. So, it was found to be not having any significant genotoxicity study effects. Ultimately, it was also found to be non-carcinogenic in a 26-week mouse study and in a 2-year rat study. So, the metabolite was found to be safe and the product was found to be acceptable. So, this is how actually the metabolite profiles are established. So, once the metabolite profiles are established, they are analyzed and if the level of metabolite is present disproportionately compared to non-clinical studies, then the metabolite needs to be separately assessed, separately evaluated for safety and toxicity studies. So, this is the reference and there is safety testing of drug metabolite guidance for industry revised by US FDA in March 2020, and besides that there are a few important papers in reputed journals related to drug metabolism and disposition. So, you can go through them for additional data. Thank you all.